Usually when we think about getting older, there's this underlying expectation of a uniform decline. Like you hit 80, your joints start to ache, your skin wrinkles, your vision gets a little blurrier.
Right.
And we just kind of assume that your internal systems are weathering at the exact same pace,
Yeah, but then you step into the world of immunology and that predictable linear timeline, it shatters completely.
Totally.
You can look at the immune profile of one 80-year-old, and they might have the robust, adaptable immune resilience of someone 30 years younger.
Wow.
But look at another 80-year-old, or even someone in their mid-60s, and they're experiencing this complete early functional collapse. I mean, the biological age of the immune system can become entirely detached from the chronological age of the person.
Which is wild. And today we're going inside that exact paradox. So for everyone listening, our mission for this deep dive is to track the frontline defenders of your body, your T cells. We're going to figure out exactly why they lose their memories, what causes that functional collapse we just mentioned, and more importantly, how cutting-edge science is figuring out how to rewind their biological clocks. So whether you're catching up on immunology for work or just want to know how your own body is aging, you're going to get the complete picture of your immune ecosystem today.
Yeah, and to really grasp the scope of this, we have to look at the two distinct roles T cells play in the body. Broadly speaking, your immune army is split into two camps. First, you have the naive T cells.
The new recruits.
Right. Think of these as fresh recruits straight out of boot camp. Their entire job is to patrol for novel, never-before-seen threats. Then you have your memory T cells.
Okay.
These are your combat veterans. They remember past infections, so like a cold you had 10 years ago or a severe flu from last winter, so they can neutralize those specific threats instantly if they ever return.
So as you age, you end up facing this really fascinating dual problem. Your immune system becomes simultaneously highly experienced, but dangerously impoverished.
Yeah.
You get incredibly rich in specific old memories, but you lose the flexibility to fight off new things.
That's spot on. The repertoire becomes severely constrained. And to understand why that constraint happens, we have to look at the production line for those fresh recruits.
The factory.
The factory, yeah. The primary factory is the thymus, which is this small organ sitting right behind your sternum. The whole maturation process for naive T cells happens here. And the data on how this organ operates over a lifetime is incredibly striking.
I read this in the source material, and it blew my mind.
It's fascinating. If a human baby is born without a thymus or has it removed very early due to surgery, they actually show only a moderate decrease in their naive T cells.
Right.
But if an older child has their thymus removed, prospective studies show they develop delayed and drastically weaker responses to novel vaccines like the tick-borne encephalitis vaccine.
Wait, so a baby losing the factory is fine, but an older child losing it is in trouble. Why the difference?
Well, it reveals how your body prioritizes building its immune repertoire. In early life, the thymus pumps out a massive diversity of naive T cells to seed the body. It's building a vast, diverse library of defenders, trying to anticipate every possible shape a new virus could take.
Oh, I see.
Once that initial massive library is built, the system relies slightly less on the constant output of the thymus to survive day to day. But when that older child loses the thymus, they lose the ability to generate specific, custom cells for a completely unfamiliar threat.
So the factory naturally winds down operations over time anyway.
It shrinks, yes. It's a process called thymic involution, and it happens in two very distinct phases. The first phase actually accelerates right around puberty. This is a growth-dependent shift.
Why puberty?
It's fundamentally an evolutionary trade-off. Maintaining a massive, hyperactive immune factory is incredibly metabolically expensive. Once you've survived childhood and built that broad repertoire of defenders, your body stops spending so much energy on maintaining the factory. It shifts those resources to reproduction and growth.
Okay, so it's like a tech startup that finishes a massive, expensive hiring spree and then aggressively downsizes the recruiting department to focus on daily operations.
That is a perfect analogy. And then the second phase is age-dependent, and this looks more like classical tissue degradation. The architectural support of the thymus, the structural epithelial cells holding the whole organ together, they slowly deteriorate and get replaced by fat and fibrous tissue.
And there's a specific trigger for this.
Yeah, a central driver of this structural collapse is the drop in a specific transcription factor called FOXN1.
Just to clarify for everyone listening, a transcription factor is essentially a protein that turns specific genes on and off inside the cell, right?
Exactly. Think of FOXN1 as the foreman coordinating the structural maintenance crew of the factory. When FOXN1 levels drop, the maintenance just stops. The architecture literally crumbles and fills with fatty tissue.
So if the factory's turning into a storage closet of fat, we aren't pumping out new recruits. How does the immune army survive for decades after puberty? I mean, we still need an immune system?
Well, the body compensates through a mechanism called homeostatic proliferation. As the overall number of naive T cells inevitably drops, the system detects the shortage. To prevent a total collapse in numbers, it forces the existing naive cells in the bloodstream, especially the CD4 positive T cells, to start cloning themselves.
But wait, if they're just cloning themselves, aren't we just making identical copies of the same recruits? Don't we completely lose the broad diversity we need to recognize a new, weirdly mutated virus?
You've hit on the exact issue. That loss of diversity is the reason older adults are far more vulnerable to novel viruses and newly emerging cancers. The body is basically prioritizing sheer quantity over diverse quality.
Wow.
And the cloning process itself causes unexpected behavioral shifts. As these naive cells are forced to divide over and over just to keep the numbers up, some of them undergo profound changes and turn into what immunologists call virtual memory cells, or TBMs.
Virtual memory, like fake veterans.
Yes, fake veterans. They acquire the functional characteristics of veteran memory cells without ever having actually fought a pathogen. They become trigger happy.
In what way?
Well, they start producing things like interferon gamma or IFN gamma and show high cytotoxicity. They act highly aggressive despite being completely inexperienced. It's basically a biological glitch caused by the desperation to maintain cell counts.
Okay, that sounds chaotic. So before we move on, let's lock this in. For everyone listening, we are at our 5-minute checkpoint. Let's do a quick recap.
That's it.
Bullet 1, the factory closes. The thymus shrinks in 2 phases, drastically cutting off the supply of new, naive T cells.
All right.
Bullet 2, the FOXN1 factor. A drop in this specific transcription factor turns the thymus into fatty tissue.
Right.
Bullet 3, the cloning compromise. To survive, the body clones existing naive cells but loses diversity. And bullet 4, virtual veterans. Some naive cells get fake memories, those TVMs, and become hyperactive without ever seeing a real threat.
Perfect summary. So that covers the recruits. But what about the actual veterans?
Right. What happens to the memory T cells that have actually seen real battle?
With the veterans, we see a phenomenon called memory inflation. Look, over decades, your immune system fights off thousands of different pathogens. But certain lifelong viruses, like cytomegalovirus or CMV, which a massive portion of a human population carries dormantly, they periodically reactivate.
And Epstein-Barr virus too, right? EBV.
Yes, EBV as well. Every single time these viruses peek their heads out, your immune system expands the specific T cells needed to suppress them back into dormancy.
So imagine a massive, beautiful library containing millions of unique immune memories. Slowly, over a lifetime, copies of just 2 or 3 books about fighting CMV start taking up more and more shelves until the other books are literally forced out into the alley.
Exactly. Because the physical space and metabolic resources of your immune system are finite, these specific viral clones hog everything. And as these cells repeatedly fight the exact same chronic viruses over decades, they differentiate heavily. They lose what we call their stemness.
Stemness meaning their ability to stay flexible and renew themselves.
Yes. For example, CD8 positive T cells shift from being stem-like into what we call TEMRA cells. These are highly differentiated cells that show profound markers of exhaustion.
What does exhaustion look like at that level?
They start expressing inhibitory receptors, things like PD-1 and TOX. Think of these as literal molecular brake pedals on the surface of the cell. They become sluggish, burnt out, and significantly less effective at clearing pathogens.
But they don't just quietly retire, do they? Because I've seen the term inflammaging popping up everywhere in longevity research. How do these exhausted veterans tie into that?
Inflammaging is a huge focus right now. It's a state of chronic, low-grade systemic inflammation that slowly builds as we age. A major driver of this condition is that these exhausted T cells develop a senescence-associated secretory phenotype, or SSP.
SAS. Okay, so what does SSP actually look like inside the tissue?
Instead of fighting pathogens cleanly and then stepping down, these old-arrested T cells sit in the tissue and continuously leak pro-inflammatory cytokines like IL-6 and TNF, as well as chemokines like IL-8 into the surrounding environment.
So they're just constantly broadcasting chemical distress signals.
Yes, and this continuous leakage damages neighboring healthy cells and creates a toxic local environment that contributes to broader systemic issues.
But think about what this means for your own body, you know? Is this toxic inflammaging just an inevitable biological clock? Or is our modern environment pulling the trigger? Because the source data had this fascinating detail comparing indigenous cohorts to industrialized populations.
Ah, yes. The population studies.
Right. And the indigenous populations have markedly different, often much healthier inflammaging profiles. It really makes you wonder how much our modern, sedentary, industrialized lifestyle is accelerating this toxic leakage.
It absolutely highlights the profound interplay between our intrinsic cellular clocks and the environment we force ourselves to live in. Though any discussion about these circulating blood cells needs a massive caveat to keep things in perspective.
Oh, the 2% rule.
Yeah.
This completely shifted how I view the immune system.
It's wild, right? The T cells circulating in your blood, which happens to be what almost all human longevity studies measure, because blood is easy to draw, make up only about 2% of your total T cells.
2%.
The vast majority are tissue-resident memory T cells, or TRM. These live permanently inside your organs, your lungs, your gut, your skin, your liver.
So we are judging the state of the entire immune system by looking at the tiny fraction currently driving on the highway, completely ignoring everyone living permanently in the cities.
Exactly. And these tissue-resident cells age on a totally distinct timeline. They adapt to their specific organ environments, slowly consolidating their residency signatures over time. They don't always mirror the extreme exhaustion or the massive memory inflation we see in the blood.
They're doing their own thing.
Yeah, they undergo progressive effector differentiation, but they are a much more stable, localized defense force, uniquely tuned to the organ they protect.
Okay, so we've mapped out the broad strokes.
Yep.
And that brings us to the 10-minute checkpoint. Let's do another quick recap for the listener.
Go for it.
Bullet 1, memory inflation. Chronic viruses like CMV cause a few T-cell clones to hog all the resources. Bullet 2, loss of stemness. T-cells shift into exhausted, highly differentiated states, those camera cells.
Yep.
Bullet 3, inflammaging and SAP. Old T-cells become toxic, leaking inflammatory chemicals like IL-6 and TNF into the body. And bullet 4, the 2% rule. Blood only shows a fraction of the picture. Tissue-resident cells age on their own unique timeline.
Perfect.
So let's shrink down and step inside one of these senescent, exhausted T-cells. We know they are tired and toxic, but what is physically breaking down at the microscopic level?
Okay, the first major failure point is at the telomeres. These are the repetitive stretches of DNA at the very ends of our chromosomes that protect the genetic code during cell division. Every time a normal cell divides, the telomeres get a little shorter.
But wait, aren't T-cells supposed to be special? I thought they had a way to rebuild those ends so they could multiply rapidly during an infection.
They do. They have an enzyme called telomerase that does exactly that. When a naive T cell first encounters a threat, it dramatically ramps up telomerase activity. It needs to clone itself thousands of times to build an army quickly, so it constantly rebuilds its telomeres to survive the massive expansion.
Okay.
The vulnerability, though, is that T cells do not reliably reactivate telomerase during subsequent immune battles years later.
I see. So imagine telomeres as a book of blank tickets. Every time the cell divides, it has to tear out a ticket. When you're young, the cell has a printing press, the telomerase, to make new tickets.
Right.
But as you age and fight off infection after infection, that printing press breaks down. You keep tearing out tickets until the book is completely empty. And at that point, the cell is legally barred from dividing ever again.
It enters a state of permanent arrest. And as it sits there unable to divide, the internal machinery starts to fail. Specifically, the mitochondria.
The power plants of the cell. How do young, healthy cells handle the wear and tear on their power plants when they divide?
Well, when a young T cell divides, it performs an incredibly elegant trick called asymmetric division. It essentially audits its internal engines, gathers up all the damaged, faulty mitochondria, and segregates them entirely into one of the two new daughter cells.
Wow.
This leaves the other daughter cell with completely pristine, perfect mitochondria.
So it forces one offspring to take all the trash so the other can thrive and carry on the fight. That's brutal but efficient.
Exactly. It preserves the energetic fitness of the lineage. But as T cells age, they lose the ability to perform this asymmetric segregation. They divide symmetrically, meaning the defective, damaged mitochondria are distributed equally into all the offspring. The garbage accumulates everywhere.
And what happens when a cell is forced to run on power plants full of defective parts?
It triggers a cascading energy crisis. The cell loses its ability to perform oxidative phosphorylation, which is the clean, highly efficient way to generate energy using oxygen. To survive, it's forced to rely on aerobic glycolysis.
And glycolysis is a much dirtier, less efficient way to get energy.
Yeah, it generates massive metabolic stress, further locking the cell into that exhausted senescent state. And cementing that state is our third failure point. The epigenetics.
Epigenetics.
Right, epigenetics is the overarching system that dictates which genes a cell can actually read and use at any given time.
It's the physical folding and structuring of the DNA itself, right?
Exactly. In aged T cells, we see profound chromatin remodeling. The physical structure of the DNA gets locked up through a process called progressive hypermethylation. By adding methyl groups to the DNA, critical genes are physically buried.
Like what?
For example, FOXO1 is a vital transcription factor that promotes stemness, self-renewal, and youthfulness in a T cell. In a senescent cell, the region containing the instructions for FOXO1 is heavily methylated. It's locked away. Same with NRF1, which impairs respiratory genes, and IL-7RA, which basically blinds the cell to survival signals.
So it's like throwing a heavy padlock on the emergency survival manuals.
Yeah.
The cell has the genetic instructions to stay young, but it literally cannot open the book to read them.
Meanwhile, other transcription factors like STAT5 actually increase, locking the cell into a differentiated, tired state. The cell becomes epigenetically hardwired to be inflexible and inflammatory.
Man, it paints a pretty bleak picture. We have empty ticket books, trash-filled engines, and padlocked survival manuals. But before we fix it, let's hit our 15-minute checkpoint to recap the molecular breakdown.
Go ahead.
Bullet 1, frayed shoelaces. T cells lose telomerase activity. Their DNA ends fray, stopping cell division. Bullet 2, broken power plants. Old cells fail to separate bad mitochondria during division, causing energy crises. And bullet 3, epigenetic padlocks. DNA methylation physically blocks the genes needed for self-renewal and energy, like FOXO1 and NRF1.
Spot on. So now the question is, if we know the exact mechanisms, how do we hack the system?
Right, let's shift to some optimism. Can we fix it?
We're getting there. There are a few layers of intervention currently being researched. The first involves chemical cleansers, specifically senolytics.
Senolytics, okay.
These are specific compounds like dasatinib or quercetin. The strategy here isn't to fix the broken cells. It's to selectively induce cell death in the zombie senescent cells. They target the specific anti-apoptotic pathways these cells use to stay alive.
So taking out the trash to clean up the neighborhood. If you destroy the cells that are leaking those toxic distress signals, you reduce systemic inflammation overall and make room for healthy cells.
Exactly. Then we have metabolic hacks. Supplementing with precursors like NMN boosts intracellular NAD+ levels. NAD+ is a critical coenzyme that acts as a metabolic sensor for energy stress.
And what does boosting it do?
When you boost it, it helps trigger a process called mitophagy, which is the cell's internal recycling program, finally clearing out those damaged, defective mitochondria we talked about. You can also use antioxidants like vitamin C or N-acetylcysteine to fix mitochondrial stress. And even exercise targets the TBK1 betaine axis to improve immune metabolism.
So you force the cell to finally sweep up the garbage engines it's been hoarding, and you fix the metabolism. That's incredible.
But the truly revolutionary interventions involve biologics and nanoparticle reprogramming. If you look at Figure 3 in the source review, scientists are exploring the use of lipid nanoparticles, the exact same delivery mechanism used in the mRNA vaccines.
Wait, really?
Yes, to deliver new genetic instructions directly into aged T cells inside a living body.
Okay, wait, so instead of extracting blood, heavily editing the cells in a multi-million-dollar lab and putting them back in, we can just send tiny lipid bubbles into the bloodstream to rewrite the cells in real time.
Exactly. We can deliver mRNA or siRNA to force the T cells to temporarily re-express those padlock stemness factors like TCF1 and FOXO1. We can use interference RNA to silence the senescence drivers. We can even deliver mRNA to temporarily turn telomerase or TERT back on, allowing the cell to print a few more blank tickets and rebuild its telomeres.
Okay, I have to throw a massive red flag on that one. If we use nanoparticles to turn the telomerase printing press back on, and we override the natural epigenetic breaks, aren't we handing the listener's body the exact blueprint for leukemia? I mean, unchecked, immortal cell division is the literal definition of cancer.
That is the most critical challenge in the entire field of immune rejuvenation, and it's a completely valid point. Safety and precision are paramount. You cannot systemically rewire the entire immune system to be immortal without courting disaster. The goal of these advanced therapeutics is transient, highly targeted modulation.
Transient meaning temporary.
Right. Imagine delivering a lipid nanoparticle that briefly rejuvenates a specific subset of T cells just days before giving an older patient a pneumonia vaccine.
Oh, I see.
You temporarily lift the epigenetic padlocks, you allow the cells to mount a youthful, vigorous response to the vaccine, and then the mRNA naturally degrades. The natural regulatory brakes re-engage.
That's surgical precision at the molecular level. You give them a temporary superpower just long enough to learn the new threat, and then you put the safety back on the weapon.
Exactly. It rejuvenates the system's plasticity without permanently breaking the delicate equilibrium between immune protection and uncontrolled cellular growth.
Man, synthesizing this entire journey. Immune aging isn't just a biological machine breaking down and rusting away. It is a dynamic, constantly evolving ecosystem. It reflects a literal lifetime of immunological learning. The body is constantly trying to balance the need to remember all the old threats with the flexibility needed to fight the new ones.
It is. It's a system that ultimately prioritizes persistence over plasticity. Our goal with these interventions isn't necessarily to erase that persistence, but to restore just enough plasticity to survive whatever evil threat comes next.
Which brings us to a final provocative question for you, the listener. Now that you understand the mechanics of your own immune aging, and you know that science is on the absolute brink of allowing us to edit our immune memories using these lipid nanoparticles, what happens if we succeed a little too well?
It's a big question.
Right. If we wipe out the exhausted memory cells and reset them to a naive state, just so we can fight off a new pandemic, what vital, deeply learned immune memories might we accidentally erase? If you reboot the hard drive to download new software, what silent, invisible protections do you lose forever?
It truly is the ultimate biological trade-off.
Take a moment today to appreciate the billions of microscopic cellular veterans currently patrolling your tissues, holding the line against a lifetime of invisible threats. Thank you for joining us on this deep dive into the battleground of your own biology. We'll catch you next time.